How can you use molecular scissors to get rid of a gene mutation? The answer is CRISPR-Cas9, clustered regularly into spaced short palindromic repeats. How do you do it? You need to get an endonuclease Cas9 enzyme. Endonuclease enzyme means that it can cut DNA from within the polypeptide chain. Okay, you need to bind it to a guide RNA. The guide RNA is basically going to tell this Cas9 enzyme where the hell it needs to go and which DNA base sequence it needs to cut, because it's not going to know on its own. So you essentially need to locate the DNA base sequence that's faulty. You need to make a complementary guide RNA that's going to bind it and locate to it. And what happens then is that you get the Cas9, it cuts the DNA base sequence, you get rid of it, and then that's the end of the story. You can either add, delete, or substitute new bases. Right, where was this concept derived from? It was derived from the mechanism of action bacteria use to defend themselves against bacteriophages. A bacteriophage inserts its viral RNA or DNA into the cell. Okay, if its RNA is going to be converted to DNA anyways through reverse transcriptase. So basically you have DNA either way. The DNA is binded to by a Cas molecule, and what happens then is it forms a spacer. A spacer is a sequence of DNA-based sequences that essentially help the cell to recognize the virus in the future. So what happens now is that a spacer is produced. A spacer is incorporated into the CRISPR array. The CRISPR array is a segment of DNA that has short DNA-based sequences repeated, and these are interspersed with spacer sequences. Okay, now what happens is that you combine the spacer with CRISPR RNA. So what happens is that it's transcribed into CRISPR RNA, and then after it's been transcribed to CRISPR RNA, you combine it with a Cas3 molecule. The Cas3 molecule basically detects and destroys any viral DNA that's complementary to the CRISPR RNA. So because we know that it's made from a spacer that's complementary to the viral DNA, so the CRISPR RNA that's binded to the Cas3 complex will be complementary to the viral DNA that will be inserted. So if that virus or bacteriophage ever in God's green world decides to enter the bacteria again, it gets absolutely annihilated. And humans have learned from this concept and started editing genes of their own.